Welcome to the 8th Annual Seattle Area.
The Seattle Epidemiologic Research and Information Center, in collaboration with the Department of Veterans Affairs, the VA Employee Education System, and the University of Washington Departments of Epidemiology and Biostatistics, presents the 8th Annual Epidemiology, Biostatistics, and Clinical Research Methods Summer Session.
Good morning, everyone. Welcome to the 8th Annual Seattle ERIC Epidemiology, Biostatistics, and Clinical Research Methods Summer Session. My name is Ed Boyko. I'm the course director. The title of today's course is Genetic Epidemiology. I'm pleased to introduce our speaker, Dr. Karen Edwards. Dr. Edwards is an associate professor in the Department of Epidemiology and the Institute for Public Health Genetics and director of the Center for Genomics and Public Health at the University of Washington. And today, Dr. Edwards will be talking on the subject of genetic association studies. Dr. Edwards.
Well, good morning, everybody. Today, we're finally going to talk about genetic association studies, which, for the most part, what I'm going to be referring to today are the case control genetic association studies. Okay? And in particular, there's a lot of information here to cover, but what I'm going to focus on for today is the pitfalls and challenges to conducting the genetic association studies. These studies are quite easy for most people to do, as many people have case control samples, and for a number of years now, many people have actually been collecting DNA, so it's quite feasible for people to conduct these genetic association studies within their own groups. And there are several pitfalls that I want to make sure that you guys avoid if you're going to conduct some of these studies. And in particular, these relate to issues of interpretation and also some basic things that you need to think about to make sure that your association study is valid. So what we're not going to talk about is the selection of cases and the selection of controls. We've talked a lot this week already about how important the phenotypic definition is, and that still applies here. In a case control genetic association study, the selection of controls is as important as it is in your traditional case control studies. And if you've had a basic epidemiology course, the same principles apply to your control selection in this situation. What we are going to focus on, though, are the caveats and the subtle differences in conducting a genetic association study and some of the issues that you need to be aware of. And those will be the things that we focus on. So again, to put this in context, and we did this a little bit yesterday, is what most of the work right now in genetic epidemiology is focusing on is the identification of these susceptibility genes that increase risk for disease. Okay? We've talked about two primary approaches that you can use to identify these susceptibility genes: a linkage approach, of which there are a variety of different methods, both parametric and non-parametric, and then genetic association studies using unrelated individuals. And as I said, it's the genetic association studies we're going to focus on today. But just a few more words about the differences, primary differences between linkage analyses and genetic association studies. As you recall from yesterday, what we're interested in looking at with linkage is the co-segregation of markers at a particular location or locus through families. Or in the case of non-parametric approaches, we're looking for sharing alleles identical by descent more often than we would expect by chance. And as we said yesterday, we don't really care what the actual alleles are. We're just using those as markers of the location. Okay? Now, when we start talking about association studies today, the emphasis shifts. We actually do care about what form of the allele at a particular locus is associated with the disease. Okay? So it sounds like a subtle difference, but it's a very important distinction that we need to keep in mind. Linkage analyses in general tend to be very powerful approaches for detecting a disease locus or a particular chromosomal region. The downfall of the linkage studies is they are not that good at discriminating between differences over very small regions. So as we talked about yesterday, you can think about linkage analysis as a pretty crude approach, in that what you'll get from a linkage analysis is the localization of a particular chromosomal region where there is likely to be a susceptibility gene. What you're not going to get in most of those approaches, unless you're doing a candidate gene type of approach, is a very precise location from linkage analysis. Okay? Your first pass through with a traditional 380 marker scan is going to give you a pretty wide region of the chromosome in which you need to follow up and investigate further. If you lay down more markers, you'll narrow that region down, but you can see that it can take several steps to get to that point. It also requires a large number of informative meioses or gametes, which means that you've got to have a lot of people, essentially. So the genetic association studies have become more appealing because the idea is that we can identify a region, a much smaller region, using these genetic association studies than we could with family studies. And I'll show you the basis behind that. Okay? But before we get there, as with most of the approaches, there are a variety of different ways that you can think about conducting a genetic association study. And the two broad categories that I've laid out here are the candidate gene studies and then the genome-wide association studies. Very similar to linkage analysis. You can either focus on a candidate gene in your association study, or you can do a genome-wide association study. And it's the same principles that we talked about with linkage analysis. If you're choosing to do a candidate gene study, it may be because you have a candidate gene whose function you believe is involved in the disease that you're looking at. If you do or choose to do a genome-wide association study, it may be because many people have tried some candidate gene studies, none of the candidate genes have panned out, and what you're saying now is, we've got good evidence that the trait is genetically influenced, there must be a gene somewhere in the genome, so let's take the positional cloning type of approach, look across the genome and see if we can't identify a region, and then look to see what genes are in that region that we may not have thought about as candidates for our disease. Okay? So again, it's the same thing that we talked about with the linkage analysis, why you might do a candidate gene approach versus a genome-wide approach. Okay? With the genetic association studies, most of the genetic association studies that we see are using a case control design, but you can certainly use a cohort study and any other traditional type of design you'd use in association studies. But the most common is the case control approach. And the basic idea behind the genetic association studies, whether it's a case control or a cohort study, is you're testing, if you're taking a candidate gene approach, whether a particular polymorphism in a particular gene is associated with your disease of interest. So now we're looking at association. So as I said, what we're looking for is evidence between, that there's evidence for an association between a particular allele and your phenotype, whatever that might be, using cases and controls. And as with all these things, before you go looking for genes, you should have some evidence that your trait is genetically influenced. But you now have a number of techniques you can use to provide that evidence. Frequently, the association studies were used as a follow-up to linkage analysis, for example, to narrow down a region that you identified in linkage, or to test specific candidate genes that were identified under a linkage peak. When you're trying to narrow down a region of interest, that's usually what we refer to as fine mapping, or as I said, to evaluate a specific candidate gene or set of genes that were identified under a linkage peak. Okay, so why should we think about doing association studies? Well, first of all, they may be more powerful for detecting susceptibility genes that have smaller effects. Now, how do you quantify smaller effects? Well, this is sort of like trying to quantify confounding. What's a confounder? Well, it kind of depends. What's a small effect? Well, essentially what I say here is with most of these things, the larger the sample size you have, the bigger effect size you can find, but it also depends on allele frequencies and a number of other factors. But generally speaking, remember that if we have a disease that is truly polygenic, where each of the genes that contribute to the variation in the phenotype contribute a small additive effect, you're not going to be able to, it's unlikely that you're going to be able to pick out one of those single genes using either linkage analyses or association studies. There has to be at least one gene that has a large enough effect that you can pick it out and see the variation in that gene. Okay? So it's difficult to quantify what is a smaller effect, but the thought is association studies will pick up genes that have smaller effects than linkage analyses will. I said association studies, we also use them quite often as a follow-up to the linkage studies to do fine mapping, to narrow down a region or look at a set of specific candidate genes. The other appealing part about association studies is that they don't require related individuals. Okay? And for that reason, they're often faster to do and also less expensive. Okay? You can generally get by with a slightly smaller sample size in terms of total number of people with an association study than you might with a family study. However, if you look in the literature, some of the estimates of sample sizes that are required in association studies are quite large. Okay. So, association studies are very appealing, as I said, for a number of reasons. Probably the primary one being many people have case control association studies that are well established, well-defined disease endpoints and outcomes. They've got DNA available, and so it's quite easy to do these. But there are a number of challenges and pitfalls in conducting these association studies, and that's what I want to spend time on today. And I've listed them here, and we're going to go through each of these in quite a bit more detail. The first issue in the association studies is that the interpretation of a positive finding and even a negative finding is not always clear. We'll focus first with a positive association that you find, let's say, with a particular marker and a disease. This was brought up yesterday. Ed asked about, if you find an association, how do you know it's that particular variant or not something right near it or in linkage disequilibrium with it? This is one of the major issues in case control association studies, is what does it mean when you actually find a positive association? Is that allele that you were looking at the actual disease-causing allele or variant, or is it something that is in close proximity or in strong linkage disequilibrium with something else that's actually the disease-causing allele? And this is something that is not easy to figure out. One of the major problems in the literature with genetic association studies is many of these are very difficult to replicate. What you'll find in the literature is the first report of a positive association with a particular allele, and then you'll see generally a series of papers that come out after that that either say, nope, we didn't see any association, or in fact, the relationship is going in the opposite direction. That what the first paper reported as a positive association, meaning increased risk, the next paper reports as a negative association and says, oh no, that's protective. Okay. So this is a major issue in the association studies, and you see it across all diseases. And so one of the major issues is that replication has proven difficult in many cases. Yes?
Why is it that, we might find a positive association in one case and a negative in another?
That's a really good question. There are a number of possible explanations, but we really, in many cases, don't know. And we'll talk about some of those potential reasons today. The one issue that reason everybody kind of falls back on is they say, well, it must be in linkage disequilibrium with the real disease-causing mutation. And we'll talk about what linkage disequilibrium is.
Well, just sort of a follow-up on that. Again, I hate to always use my example of Alzheimer's disease and this ApoE gene, but one of the thoughts is the ApoE4 gene was the original gene and may have had some major beneficial effect over 300,000 years ago, and as things have changed, and as people are growing older now, it's got a deleterious effect in older people. So benefit may be in one circumstance and deleterious in another, depending on your circumstances. And it seems to me that really trying to define what the gene is and what all of its possible phenotypic outcomes are is really a critical issue also.
Yeah, I think that's a really good point, that we have to be careful in calling something a susceptibility gene or a bad variant, because there are some very good examples of things. Let's take sickle cell anemia and malaria. People with sickle cells are actually protected against developing malaria. And if you think about where these things are endemic, it kind of makes sense that these particular forms of a gene would develop over time and become more constant. There are a number of other examples like that. There's a lot of interesting stuff around obesity right now and the thrifty genotype hypothesis, a number of different things. So I think, we're looking at this at one point in time and in our current environment, but we have to think about the evolutionary history of populations and keep in mind that many times these things that are deleterious in our current environment may have actually been something related to survival in different environments. So we've got to be really careful, especially as people start thinking about, gene therapy and a host of other issues which we won't go into. We just don't know enough, and I think it's important to keep that in mind because there's some really interesting things out there. Okay. The other thing that we'll just mention briefly about genetic association studies, which really this is no different than a traditional case control study, is the issue of power in detecting interactions. And in particular, what we're interested in genetic epi are gene-environment interactions and gene-gene interactions. We're no better off in the genetic association environment in terms of power to detect interactions. And maybe even in some cases, a little worse off, because for some of the genes that we're interested in, the variants can be very rare. And so it's really going to be difficult if the variant that you're interested in is quite rare. It'll be very challenging to pick up gene-environment interactions. Okay? And there have been a number of newer study designs which might help a little bit. Some of these are the case-only studies, which, if you're not familiar with, those are an interesting approach to think about using for evaluating interactions. You can only look at the interaction in these particular situations, not main effects, but it might be a nice supplement to a study if you look into that literature. So case-only studies. We're not going to spend a lot of time on that, though. The other major issue that we have to think about in the genetic association studies is confounding. And again, no different than traditional case control studies, except in genetic studies we have some very particular instances that we have to consider. So we're going to spend a fair amount of time today talking about this. And then, of course, multiple testing. In most genetic association studies, well, up until about this point, people for the most part have looked at candidate genes. Now, it may be 1 to 20 candidate genes, and then within those candidate genes, you may have multiple markers that you consider. And so we certainly are running into problems with multiple comparisons at that point. But we are in a stage right now where the multiple comparison issue is going to become very problematic, because people are now talking about doing genome-wide association studies in the case control setting using 100,000 or 500,000 SNPs. So the multiple comparison problem has just gotten very, very large. And what makes it even more challenging is that the markers that you're looking at are not independent. They're correlated with each other. So the correction for multiple comparisons is not straightforward in this setting. If you were to use something like a Bonferroni, that is very conservative because it assumes that your tests are independent. And so it overly adjusts for these multiple comparisons. There are some new methods out there called false discovery rate methods and extensions of that, which if you're considering doing a genome-wide association study, you should take a look at. Because this issue of multiple comparisons is a major limitation of doing these large association studies, meaning large with lots of genes or lots of markers, and is something that you can't ignore, but it's not straightforward to deal with. Okay? There are a variety of approaches in the literature to consider. And we're not going to go into them today, but I'm just going to tell you that it's a critical issue that you need to think about. Okay. Okay, so let's talk a little bit about some of the possible reasons you might observe a positive association between your marker allele and your disease outcome. It is possible that the marker you have selected is actually, for example, the functional variant and is part of the disease process. That's actually a good thing. That's what you'd like to have. And if that were the case, you might expect, although this does not always hold, is that you would like to see the same positive association in at least several different populations. Okay? That would be something that you hope could be replicated across different groups. If you actually have a variant that is a functional variant, changes a protein structure, changes an enzyme level, makes some change that you can clearly see would be related to your disease outcome, you would hope to see that same positive association in several different groups. So that sort of is our gold standard. The other distinct possibility is that the marker that you have chosen as the marker of interest is not actually the disease-causing variant, but is in let's just say it's correlated with the actual disease-causing variant. It's in linkage disequilibrium with it. And so in that situation, the true functional variant has not been measured, but you've picked up the association with your marker because it's highly correlated or in linkage disequilibrium with the susceptibility variant. Okay? And this is the one that we're going to spend some time talking about today, is what is linkage disequilibrium? How does it help you? In some cases, how does it blur your ability to interpret your results from an association study? The other possible explanations, as I said, for observing a positive association is confounding. And we're going to talk about this one in a lot more detail. In genetic association studies, we have a unique situation that really gives us some pause when we think about confounding. And in this particular situation, race and ethnicity is the confounder we're most interested in. And I'll tell you why that is. It's something that relates to population stratification, which we'll define in a minute, but is probably a term that you've heard if you're looking at the genetic epi literature and looking at case control association studies. It's also possible that there are other genetic and environmental factors that might result in population stratification. So we'll define what population stratification is, and then you'll understand how these other factors can cause this particular form of confounding. And then, of course, chance is always a possible explanation for a positive association finding, but hopefully what you've done is dealt with the multiple comparison issue up front so that you've at least decreased the possibility that your result is due to chance alone. Okay, so population structure and population stratification. As I said, these are terms that you will see in the literature if you're reading any genetic association studies, and is something that you need to be aware of when you're conducting your own association study. So let's just look up here at the example under A. This is the classical definition of confounding that, as epidemiologists, we're all aware of. So what you're trying to do in your association study is look at the relationship between the exposure and the disease and determine whether or not your exposure is related to your disease. However, we know that there are factors that, if they're related to both the exposure and the disease, that if we don't control for those factors, what we may see is a positive, what looks like a positive association, but that is really due to the confounder. Okay? So in some ways, the confounder makes it look like there's a true association when in fact there's really not. Now it can go either way. It can either mask a true association, or it can make it look like there's an association when there's not. In genetic association studies, what we see is that ancestry is our confounder of interest here. And the reason for that is, as you'll see in a few minutes, that if you think about different groups around the world, different population groups around the world who may have developed in isolation, let's just take different continental groups. What you have is that within each of those different groups, based on whatever the founding population was, you will have certain alleles found in that group. Those alleles may be, particular alleles may be at higher frequency in one group than in another. And what we see very often is that at different genes, the marker alleles have very different prevalences in different populations, to the extent sometimes that particular alleles are only seen in one group and not others. So all groups have the same gene, but it's the markers, the alleles at those genes, that the prevalence can vary dramatically. Okay? And then if you add to that, because remember confounding, we have to have both situations occurring, that the confounder has to be related to the exposure. In this case, we've said that we know the genotypes can vary dramatically across different groups. We also know that disease prevalence can vary dramatically in different groups. Let's take diabetes. The prevalence of diabetes in different groups varies tremendously. In a Caucasian population, the prevalence may be about 6%, whereas in Native American populations, it can be as high as 50% or higher. Okay? So if you take that example, and you say that in a Native American group and a Caucasian group, we know the frequency of diabetes is tremendously different. We also know that the marker alleles at different genes vary dramatically between Native American populations and Caucasian populations. If you, let's just take the most extreme case, had a sample of individuals who were Caucasian and a sample who were Native Americans, you put them together as one case and one control group, because of the difference in both diabetes and the prevalence of particular diseases, you may see a spurious association. And in particular, what you're likely to see is whatever, let's say it's your candidate gene, whatever marker is in the highest, essentially when you put those two groups together, you may see a spurious association, and I'm going to show you an example in a minute, where it looks like a particular gene or marker is associated with the outcome, but it's due entirely to confounding. That when you stratify the sample and look separately in the Native Americans and separately in the Caucasians, there is no relationship. But when you put the two groups together, you see a spurious association. So that's the most extreme case. It gets more complicated than this because many individuals are not of a homogeneous ethnic background, and there's something called admixture. So for example, in many groups, there is a mixture of Caucasian, Hispanic, many other ethnic groups. And if you sample from your population such that your case group is different than your control group in terms of in terms of ancestry, you can also induce spurious associations. This is quite a challenge because, as I said, in most cases, people will not at this point put different groups together because of the potential for population stratification and analyze as one group, unless you know that the conditions for confounding are not met. And the only way that would be true is if the disease prevalence did not vary or the allele frequencies did not vary between different groups. But even still, you're going to want to do a stratified analysis. With the admixture, as I said, it's a little bit more challenging to think about, but there are some new statistical methods that we can use to try and get at this, and I'll mention those today. Yes?
Well, because of the genetic evidence that has been established at by several people that migration patterns out of Central Africa kind of have been established as the origin of lots of population. How long does it usually take for these markers to get established? Is it a matter of 1,000 years, a matter of 10,000?
Yeah, that's a hard one to ask. I'll say it depends. It's kind of like confounding. It depends. What you'll see, and actually this slide, we may be able to address this. This also, though, has implications for things that we're going to talk about tomorrow, is that frequently Africans have a much more diverse genetic makeup than other populations because if you think about it, that population is much older. There's been more time for diversity. There's been more time, for the markers to be more polymorphic is what we see. There are more polymorphisms. There's more variation in African populations than in others. And this has really important implications when you're doing these genetic association studies. I mean, this is one of the key and critical issues that we have to keep in mind as we're doing these studies. And what this figure shows, this is just a very nice figure, that shows if you look at these circles, we can assume that these circles are a particular candidate gene, and each little section of the circle is an allele. And if you look just at the kind of the proportion of the colors across these different populations, you can see that there's quite a bit of variation. So I think you can see this kind of purplish color the best. So if we look right here, you can see that this purple color is quite common in this particular group. Whereas if you look over here at a different group, there's a much smaller proportion of that color. So essentially that allele is much less common in this group than it is in this group. And you can see very clearly from these figures that the allele frequencies vary dramatically across these populations at any particular gene. Okay? As I said, this is well established and something that we have to keep in mind when we're doing genetic association studies. Okay? Yes?
I was thinking about what you were talking about yesterday in terms of heritability, and that it would seem like also that it would depend a lot on the population that you might choose. That, I mean, in some ways, diabetes might have a lot higher heritability in one population than in another, and that may be a consideration, that you choose a population that might have the higher heritability.
Right. That's a good point. So it could be that the heritability is higher because maybe the genetic effects are stronger, but it can also be the environment. So we have to be a little careful with the heritabilities. But you're absolutely correct in that when we think about trying to identify these genes, the choice of the population is very important. And this is why you'll see in the literature, up until very recently, kind of the mantra was we want homogeneous populations where the disease is present. Okay? So with diabetes, there have been a number of studies done in the Pima Indians. There have been a number of studies done in the Finnish population because you think about Finland and some of the Scandinavian countries, they're small, they're isolated, they're old populations, and people thought of those as very homogeneous groups. What we're finding now when we look at these genetic markers is they're not as homogeneous as we thought. And so this concept of a homogeneous group, I think, it's not going to be long before we're not saying that anymore. Okay? We're not going to be saying particular groups are homogeneous based on, the characteristics that we're able to look at. We may need to be defining homogeneous groups based on genetic information, because it was eye-opening when we looked in Iceland and showed that what we thought was a very homogeneous group, we actually can see evidence for population structure even in that group. So it's fascinating. So we have to be careful that we don't assume that a group is homogeneous, and what do we mean by homogeneous? So it's a really interesting area, and there's a lot of fascinating stuff in the literature out there if you're interested in looking into this. But it's just, these are kind of the pitfalls and the things that the assumptions that we've made for a long time, that as we start to look at this, it's like, well, this is interesting. Okay? Groups are not as homogeneous as we thought, and it's, you can think about the population history and diversity, and it does make sense. But again, our assumptions aren't always correct. So this actually is a paper from Knoller published in 1988. This is the classical paper that first made us aware that confounding by admixture may be a really important thing to think about when we're doing these case control association studies. So I can't quite cover up pieces of the slide here, but let's just first look at the relationship in the overall population. So this was a sample of Pima Indians. And what they were looking for, as I said, back in 1988, we didn't really look at haplotypes in the same way that we are now. But let's just pretend that what they're calling this GM haplotype is a candidate gene and a particular marker, so a particular allele. So they were showing that the overall frequency of this particular allele in the overall Pima population was 6%. Then what they're looking at is the frequency of diabetes, the percent of subjects who have that allele. The frequency of diabetes in subjects who had that particular allele was 8%, versus the frequency of diabetes in those who didn't have the allele was 29%. So in fact, what this would show is that this particular allele was protective. It was associated with a decreased risk of having diabetes. Everybody got very excited at first, but fortunately, Knoller's group looked at this a little more closely. And what you can do is by looking at the number of grandparents who are either full Pima Indian or Caucasian, you can stratify that population by degree of Pima ancestry, depending on whether they have 0, 1, or 2, or 3 Caucasian grandparents. Okay? And so if we do a stratified analysis, what you see is the frequency, first of all, of this allele varies dramatically by how much Pima ancestry you have. So for example, in individuals who had no full-blooded Pima ancestors, the frequency of that allele is very high. And in those who had 4 Pima grandparents, the frequency is very low. So here we can see that we are set up for confounding. The allele frequency varies dramatically by degree of Pima ancestry or degree of Caucasian admixture, and we know the prevalence of diabetes does too. And so what you see if you look at the stratified analyses, that there is no difference in the frequency of diabetes with this particular allele. This is the classical case of confounding, and in this case it's due to admixture with Caucasian ancestors. Now, for the most part, people would consider a Pima population a relatively homogeneous population, This is why so many studies on diabetes have been conducted in this group. But you can see that it's very easy to fall into this trap and have what would be a pretty strong spurious association if you did not take admixture into account. So it was really this paper that kind of lighted the fire that we need to think about this and figure out what was going on. As I said, this was back in 1988. We didn't have the number of genetic markers we have now. We didn't fully understand the diversity and genetic diversity of populations like we do now. So this was a classic paper. It's referred to frequently in the literature, and I would encourage you guys to go back and read this paper. It's, as I said, quite dated, but it's a classic and it's a very good paper. And I think it will really help you to understand this issue of population stratification. It's laid out very nicely. This paper may kind of represent the extreme side of confounding, because in this particular example, the prevalence of diabetes varies so dramatically between a Pima population and a Caucasian population, and so does the allele frequency. So this may be an extreme example, but it just shows you how important this could actually be, okay? And may be relevant to many other diseases we're looking at today as well. Okay, so you can see why we're so concerned about this issue of population stratification. So how do you deal with it? Should you just forget case control association studies, period? Or are there things you can do? Well, as I said, the methods to deal with this include restricting to a homogeneous population. I think we can probably check this one off the list, but let's put it in perspective. If you have different ethnic groups where you know that the allele frequencies differ and the disease differs, that would not be a homogeneous group for your case control study. Okay? There is some thought that if you were to restrict, for example, to either a Caucasian population or an African or an African American population or an Asian population, that maybe you're okay. But there's actually now evidence that that is not the case. Okay? Particularly in African American populations, also in some of the Latino populations, and possibly even in Native American populations. Caucasian populations we're thinking that that is not true either, but the methods to identify structure within Caucasian populations people are working on. So it's not straightforward, but I'm guessing in the next year or two you'll see a number of papers coming out suggesting that it's an issue in these populations as well. Okay. So then, given that that's, yes?
If I could go back to the previous example on population stratification for just a minute, please. If we look at the percentage of Pima Indian ancestry, the groups with 100% ancestry have much higher prevalence of diabetes.
Yes.
So than those who have none or 50% ancestry. So is this a case of linkage disequilibrium, so that the particular gene that was examined was not, or the particular haplotype was not associated, but something that's closely associated with it might be?
Well, I think what this is showing, if we were to lay this table out a little differently, what this percent of diabetes shows is that the prevalence of diabetes varies by ancestry. So what we can see down here is that the prevalence of diabetes in those subjects who had 4 Pima Indian ancestors is very high, whereas the prevalence of diabetes in those who had no ancestors was very low, okay, for grandparents of pure Pima ancestry. So what that shows us is kind of what we already knew, that the prevalence of diabetes is very different in a Caucasian population versus a Pima Indian population. And then what this shows is a similar phenomenon where the allele frequencies vary depending on your ethnic background. Now, what gives us concern is not so much that these, the percentage of diabetes varies in these different strata, but the fact if you were to combine all subjects together, you get a spurious association. Okay? And back in 1988 and beyond, we didn't have as many candidate genes as we do now, and we certainly did not have as many markers. And so there were a number of genes that were looked at across a variety of populations. One of the classic is HLA haplotypes and genotypes. We've known about HLA variation for a long time. HLA was probably one of the genes that we've studied the variation of from very early on and looked at HLA markers with different diseases. And so at this point in time, we didn't have that many candidate genes to look at. We didn't have that many markers to look at. And so I don't remember exactly what this particular marker was, but I seem to recall it was something like an HLA type of gene. So, although it is entirely possible that this was not the disease-causing gene, I think the more important message here was the fact that you could get a spurious association if you didn't take into account this admixture. And as I said, it was really the first paper that demonstrated this issue, and just a very nice classic paper. And I think that because this was such an important message, nobody really remembers what the primary point of the paper was, that this has become the focus of the paper. And I actually have to admit, I don't remember what the GM haplotype was, but I think it was an HLA type of marker. Okay. Does that answer your question? Yeah. I mean, it still could be an issue with linkage disequilibrium, but the basic problem is that there's no association if you look in the stratified sample. Yeah. It could just be that you've got the wrong marker, too, which obviously there's no association with it. Okay, so since we still need to think about making sure that our study, our cases and controls, that we're not adding to the problem by putting different groups together where we know we could induce population stratification. But we have to be a little careful about what we're calling a homogeneous population now. Just because you restrict it to one ethnic group does not guarantee that you're not going to have problems, because even within a particular racial or ethnic group, it's still very possible you're going to run into problems with confounding or population stratification due to admixture. Okay. And with some populations, it's been very well studied, and we know that you can't just assume that you're going to be okay. So another approach is to use, there are a number of family-based studies that actually are family-based association studies, which we're going to talk about next hour, that are not sensitive to population stratification. So that's another option, but then you're back to needing to collect family data, although more limited than what we described for linkage studies. It's also possible that you can try to adjust, as you would in a traditional epidemiologic study, to adjust for these confounders. Our choices here, though, again, are a little problematic. Up until very recently, we would use information on self-reported race or ethnicity to try and adjust. And in some situations, that will help you a little bit, but it's a problematic variable. We'll just leave it at that. What has emerged more recently, though, are statistical methods to deal with this issue of population stratification using genetic markers. And there are two primary approaches, and of course now with each of these there are variations on the theme. But generally what you will see is discussion about two approaches, genomic control and structured association, as methods to use genetic markers to help you adjust for population stratification. So I'm not going to go into a lot of detail about these, but you should be aware of them. There are a number of resources out there in terms of software that will help you to do this, and there is a lot of literature out there on these methods. So genomic control, the idea here is that what you're doing is adjusting your chi-square statistic for this over-dispersion of the test statistic. There is software out there to help you accomplish this. However, this is probably not the choice of most epidemiologists, okay, but certainly has been used. It would be a long discussion to talk about why you might, in which situation you might use genomic control, in which you might use structured association. But as I said, there are a number of review papers out there in the literature. If you really start to do these association studies, you should take a careful look at the literature and help you decide which of the approaches best fits your data and your situation. Something that's a little bit more familiar to epidemiologists is the structured association approach. And essentially what you're doing with this approach is you're using genetic markers to help you identify underlying populations. And it's using the concept that different subgroups are going to have different allele frequencies, and that you might be able to use genetic markers, ones that are not related to the disease you're looking at. So this is why we say unlinked markers. They should not be ones associated with the disease you're looking at, but random markers throughout the genome. And using that to try to identify subpopulations within your sample. And then essentially what you do is a stratified analysis based on the subpopulations that were identified using these genetic markers, unlinked genetic markers. So to epidemiologists, this feels familiar to us. It's like a stratified analysis. We can understand it. And again, there's software available also to accomplish this type of adjustment. Okay? Both pieces of software are out there. And as I said, there are some newer twists on both genomic control and structured association. But these are the two basic approaches. Now, we spent all this time talking about population stratification, and I should tell you that in the literature and even among people working in the field, there is debate as to how important this really is. As I said, I think the Knoller paper kind of gave us the warning signal that we need to pay attention. But some people have suggested that maybe this isn't as big of a problem as we think it is. And I think where this debate came from is some people were suggesting, we shouldn't be doing these case control association studies because of this potential problem. And then people have weighed in and said, we don't think it's that big of a problem. And others said, well, we do. So I'll just let you know that there's an active debate out there about how important this problem is. And I think the thing to keep in mind is that it depends. It depends on the disease you're looking at. It depends on the gene you're looking at. If you're in the situation where we saw with Knoller, where the allele frequencies vary dramatically across populations, the disease prevalence varies dramatically across populations, and your population is one where there is admixture or some other sort of structure, you're going to have problems. So I think that's kind of what you guys need to think about when you're evaluating your particular studies or designing a study. Make sure that you're not inducing or making the situation worse. And there may be some situations where you're going to be okay. So what I have on these next two slides are kind of the point and counterpoint on the debate about population stratification. And it's probably worth taking a look at these. This particular study seems to think that it's a serious issue, whereas the other side thinks that, it's probably not that big of a problem, but we need more evidence, especially from Caucasian populations of European descent. This is where it gets a little bit more challenging. We certainly know that within Latino populations and African American populations, you can't ignore this. Okay? So my take on the issue of population stratification is I think that you need to be aware of it and you need to look for it. You need to design your study as carefully as you can to make sure that you avoid this, as I said, to the extent that you can. In some situations, I would say in most situations, this does not strike a fatal blow to the case control association study, especially now where there are methods like the genomic control method or the structured association method where you can adjust for this if it exists in your sample. But the problem is you have to have these unlinked markers to make sure that you can adjust. And the issue with having the unlinked markers is we don't actually know how many markers you need to have. The estimates vary anywhere from 25 to 500 markers. When you're writing a grant or you're doing these case control studies, 25 or 500 markers is a big difference in the budget. Okay? The other issue is picking these markers is challenging because what you want are markers that show marked allele frequency differences across different populations. If you pick markers where there's no variation in the allele frequency, it's not going to help you pick up this underlying structure. And so then what you have to know is what is the allele frequency in each of the different populations which you think is contributing to your sample. So if you've got a Caucasian sample of European descent, do you have information on allele frequency gradients from north to south and east to west? Those are going to be the markers that help you determine whether or not you've got structure. Okay, so it's not straightforward, but there are at least methods that we can use to deal with it. And so when you're designing a study and thinking about doing genotyping, you need to think about including these unlinked markers in your sample. There are also another class of marker which we call ancestry informative markers, or AIMs. And at least for some African American populations, a set of AIMs has been identified that you can use to detect admixture within those groups. And in that particular case, you may be able to get by with as few as 24 or 25 markers to detect structure. But as I said, with some of the other population groups, we don't know yet which markers, how many markers, and I said especially if you're dealing with a Caucasian population. Okay. So these are, you can see why I say there are a number of challenges and pitfalls in conducting these association studies. But you shouldn't give up. There are methods, you just need to think about them when you're designing your studies. Yes?
I guess I favor the counterpoint view here. I think that if you look at an allele and it's associated, I think that's progress, even though the allele may not really be causally related to the outcome. It means that you've identified something which indicates something else, and in a really general sense, I think that I guess you shouldn't go out in making preventive or treatment recommendations on the basis of that discovery. I think it's got to be accepted cautiously and subject to more scientific scrutiny. But it seems like it's progress to me rather than whatever the reverse of progress is.
Yeah, well, I'll say a couple of things. It could be, and I think, as we're doing these association studies, we just have to be aware of the interpretation that when you find a positive association, don't run out and say, I have the variant that causes the disease. Consider your other alternative explanations and, make sure that to the best you can, as you would with any case control study, you can at least rule out or understand how those alternative explanations may play out in your sample. I would say the only disagreement, slight disagreement I would have is that with a lot of these association studies, we can't replicate them. And so we don't know, are they a spurious finding? For example, in the Knoller paper, that marker was not associated with the disease. Okay, so, and we couldn't even say that it was in linkage disequilibrium with something else. It was a spurious association that when you do a stratified analysis, it wasn't there. Now, what you may be seeing, at least with that particular marker, is it's, a marker that would tell us about degree of, ancestry. So that may be informative. There may be some situations where you see a positive association and it's not the causative marker, but it's in linkage disequilibrium with something else. I think that's informative. The problem is it's hard to tell what's spurious due to population stratification and what is a positive finding and is in linkage disequilibrium with something else. And I think what people are concerned about is, for those spurious associations, how much resources and time are we putting into following up on things which are spurious versus the things that are in linkage disequilibrium, which actually are moving us forward. And that's the hard part, and that's where we can't tell. So I think that's why designing your study as best as you can is really important, so you don't spend a lot of time and resources trying to replicate something that was a false positive because of, not necessarily bad design, we don't have all the information about this population stratification. So it's really quite challenging, and I think there's no good answer, but just when you find a positive association, think about the alternatives, and of course you should publish it and pursue it, and then try to, the best you can, figure out whether or not it's real, in linkage disequilibrium, or a false positive. So it's worth doing the association studies and makes them quite interesting, shall we say. It'll keep you up to speed on methods. Okay? So a couple of things, other things that are important in the genetic association studies, moving beyond these major issues now, is back to another more basic issue and something that, again, is unique to doing a genetic association study, is now we're thinking about the marker alleles. And because these are genetic markers, one of the things that we can look for is to evaluate whether or not our markers are in something called Hardy-Weinberg equilibrium. In other words, given the allele frequencies and given what we know about populations, we should expect to see genotypic classes at a certain rate. Okay? If in your sample your marker is not in Hardy-Weinberg equilibrium, that may indicate problems with either your sample collection, it may indicate problems with genotyping errors, or it may indicate something odd with your marker. Okay? And in particular, when we talk about Hardy-Weinberg, we really are talking about, if we're doing a case control study, evaluating whether or not the markers are in Hardy-Weinberg in the control group. Because if you think about it, if you've selected a candidate gene because you believe it's involved in the disease process, if it truly is, you would expect the allele of interest, whatever that disease-causing allele might be, to be at higher frequency in your case group than in your control group. If that is the cause of the disease, that's what you would expect. And so if you look at Hardy-Weinberg in your case group, you would actually expect to see deviations from it because you are hoping that you've got a higher frequency of a particular allele among your cases, if that's the one associated with the disease. But in your control group, that should represent your population, your underlying population, and you should see Hardy-Weinberg in your control group. If you don't, you need to exclude that marker from your analyses in many situations because it violates one of the assumptions, and again, it may lead to a spurious positive finding when it really isn't there. So Hardy-Weinberg is something you need to check for all of your markers in conducting these studies. So again, let's just summarize some of the potential reasons why you might see a deviation from Hardy-Weinberg. One possibility is that there's laboratory error in calling your alleles. If you find that your marker alleles are deviating from Hardy-Weinberg, the first thing that you should do is go back and check your laboratory results. There can be other issues which you can't do anything about, like non-random mating in the population. So for example, assortative mating. Maybe the gene that you're looking at has some sort of effect on survival. Maybe you're not going to see a particular genotypic class in your population. Okay? Migration, mutation, genetic drift, and then of course sampling. You may have just sampled, especially if it's a small sample, from a particular group of subjects with a particular genotype. Just bad luck. Okay? The one other thing to think about when you're doing an association study is you have the choice here of how to define your exposure. And you're thinking about your exposure as your candidate gene. At your candidate gene, you can either look at the allele or the genotype as the exposure of interest. If you're using the allele as the genotype, this can be appealing because essentially you double your sample size. Everybody has 2 chromosomes, therefore they have 2 alleles. The problem is people have genotypes, and so the interpretation of an analysis based on alleles, again, can be a little bit challenging because we all have genotypes, not just one allele. We have 2 alleles. As I said, departures from Hardy-Weinberg equilibrium can affect your error rates if you're not in Hardy-Weinberg, but fortunately there are some corrections for this, but it's a little bit challenging. Most people will just throw out a marker if it's not in Hardy-Weinberg equilibrium in their control group. Okay? So that concludes our session on association studies for today. Thank you.